Imagine a bustling city surrounded by a highly intelligent border. This border decides who can enter, who must leave, and what should stay inside. In every animal cell, that border is the plasma membrane, also called the cell membrane. So, the first question is, what is the plasma membrane? The plasma membrane forms the boundary between the cell and its external environment. Although it's extremely thin, it performs several critical functions. It protects the cell, regulates the movement of material, allows cells to communicate with one another, and helps maintain the stable internal condition necessary for life. One of the most important ideas in biology is that the plasma membrane is selectively permeable. Yes? So, what is the meaning of selectively permeable? This means some substance can cross easily, some substances can cross easily, while others are restricted or require assistance. Yes? Others are restricted or require assistance. Yes? So, according to the fluid mosaic model, the membrane is not a rigid wall. Instead, it's a dynamic and flexible structure. The membrane is primarily composed of a phospholipid bilayer. Yes? Each phospholipid has a hydrophilic or water-loving, yes, head and two hydrophobic or water-fearing fatty acid tails. In an aqueous environment, the phospholipids arrange themselves into the two layers. The hydrophilic heads face the water inside and outside the cell, while the hydrophobic tails point inward toward each other. This arrangement creates a stable barrier that separates the cytoplasm from the extracellular fluid. The membrane is called the mosaic because many different molecules are embedded within it, especially proteins. Yes? Membrane proteins carry out most of the membrane's specific functions. Some proteins span the entire membrane. These are called integral membrane protein. Others are loosely attached to one side of the membrane and are known peripheral membrane protein. Yes? These proteins may serve as transport channels, yes, carriers, enzymes, receptors, for signaling molecules, anchors connecting the cytoskeleton to the membrane, or recognition molecules that help cells identify one another. Without membrane proteins, cells would be unable to communicate effectively or regulate the movement of many substances. So, animal cells membranes also contain cholesterol. Yes? Cholesterol acts as a regulator of membrane fluidity. Yes? Acts as a regulator of membrane fluidity. Yes? At a high temperature, it restrains excessive movement of phospholipids, while at lower temperatures, it prevents the membrane from becoming too rigid. As a result, cholesterol helps maintain the proper consistency of the membrane under changing conditions. So, we can write cholesterol helps maintain the proper consistency of the membrane. Yes? So now let's discuss how substances cross the membrane. Yes? So the first major category is passive transport. Yes? Passive transport doesn't require the cell to expend energy. Yes? Doesn't require the cell to expend energy. Yes? Substances move down their concentration gradient from regions of higher concentration to regions of lower concentration. Yes? Higher concentration to lower concentration. The simplest mechanism is simple diffusion. Yes, this method, simple diffusion. A small non-polar molecule such as oxygen or carbon dioxide. Yes, oxygen or carbon dioxide can pass directly through the phospholipid bilayer. For example, oxygen diffuses into cells where it's needed for cellular respiration, while carbon dioxide diffuses out as a waste product. No transport protein and no ATP are required. Yes? So, a special type of diffusion is osmosis. Yes? The diffusion of water across a selectively permeable membrane. Water moves from a region with lower solute concentration, yes, with higher solute concentration, either directly through the membrane or through specialized proteins called aquaporins. Yes, understanding osmosis is easier when we compare three solutions. In a hypotonic solution, the surrounding fluid has fewer solutes than the cell. Water enters the cell, causing an animal cell to swell and potentially burst. In an isotonic solution, the solute concentration is approximately equal inside and outside the cell. Water moves in both directions at similar rates, and the cell remains stable. In a hypertonic solution, the surrounding fluid contains more solutes than the cell water leaves the cell, causing the cell to shrink, a process called crenation. Maintaining the correct osmotic balance is essential for the survival of animals. So, some molecules cannot cross the lipid bilayer easily because they are large or charged. In these cases, the cell uses facilitated diffusion. Yes? Facilitated diffusion still moves substances down their concentration gradient and requires no ATP. Yes? No ATP. But it depends on transport proteins. Yes? These proteins. There are two major types. Channel proteins bind to a specific molecule, channel proteins form hydrophilic tunnels through the membrane. Ion channels allow specific ions such as sodium, potassium, or chloride to pass. Carrier proteins bind to a specific molecule, change shape, and release the molecule on the other side. Glucose transports are classic examples. Both mechanisms increase the efficiency and selectivity of transport. So, the second major category is active transport. Yes? Active transport requires energy because substances are moved against their concentration gradient from lower concentration, lower concentration to higher concentration. Yes, ATP requires. The energy usually comes from ATP. The sodium-potassium pump, for example, the sodium-potassium pump, one of the most important example. This protein uses ATP to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. This process creates electrochemical gradients that are essential for nerve impulses, muscle contraction, and many other cellular functions. The sodium-potassium pump demonstrates how membrane proteins can perform complex tasks using cellular energy. Cells can also perform co-transport. Yes? Sometimes called secondary active transport. In this mechanism, the energy stored in the gradient of one substance is used to move another substance across the membrane. For instance, the movement of sodium ions into a cell can help transport glucose into the cell at the same time. Yes? So, now we can talk about bulk transport. Bulk transport. Moving large materials. Some materials are simply too large to pass through channels or carriers. Cells solve this problem with bulk transport, which involves vesicles. Endocytosis brings substance into the cell. Yes? Yeah. Into the cells. During endocytosis, the plasma membrane folds inward, surrounded the materials, and pinches off to a form of a vesicle. Yes? There are several forms. Phagocytosis. Yes? Phagocytosis. Or cellular eating. Cellular eating occurs when cells engulf large particles or microorganisms. Certain white blood cells use this process to destroy pathogens. Yes? Destroy pathogens. Pinocytosis or cellular drinking takes in the droplets of extracellular fluid. So pinocytosis meaning is cellular drinking. Yes? Receptor-mediated endocytosis is highly specific. Yes? And C, receptor-mediated endocytosis is highly specific. Molecules bind to receptors on the membrane, and the membrane then forms a vesicle containing those selected molecules. Yes? The opposite process is exocytosis. A vesicle inside the cell moves to the plasma membrane. Yes? Fuses with it. Yes? Fuses with it and releases its content outside the cell. Yes? Exocytosis is used to secrete hormones. Yes? Secrete hormones. Yes? Neurotransmitters. Neurotransmitters like acetylcholine, digestive enzymes, and many other substances. This process is also important for adding proteins and lipids to the plasma membrane itself.